Yeah, so I'll go right in. So as it was introduced, I actually work mainly, my core work is actually in juvenile diabetes research or type 1 diabetes. So it may seem a little bit left field, but hopefully it makes sense by the time you get uh, through the talk. Whoops, sorry. So the contents I'll go through very briefly on the background, and then sort of how we've been using twist capture sequencing for human virome research, but also clinical diagnostic, informing that, and sort of give you a flavor of a few case studies that we've had recently, and current state of clinical metagenomics in Australia. I'll kind of touch upon that towards the end. Some barriers to implementation, what I think, and then sort of future directions, and a bit about sustainability at the end. So I think there were a lot of international speakers. So I mean, not speakers, so attendees. So I just wanted to make sure you guys know who we are down under. We're sort of population a bit less than the state of California. Sort of the major states I've broken down there, the three main populated. And then obviously we've got it's a massive continent, so we get various climates and so various viral bacteria outbreaks and some tropical conditions up north. And then we got the other spectrum, so south is quite cool. And then so it's a melting pot of international travelers and multicultural, diverse. And then we've also got the indigenous communities and rural communities throughout the continent. So we do a lot more science rather than just Vegemite and Tim Tams and so forth. And I guess what's relevant to in the space of clinical metagenomics and diagnostics is that we have a Medicare system. So that's a federally funded medical care system. A lot of these diagnostic tests, we hope that it will become included as part of the Medicare, so it's less out of pocket from the people that need to access them. So yeah, I won't go too much into that. So my background, I mean, I actually have various switching of fields and expertise. I actually did my PhD at Sydney University, working on small RNAs, and it was actually in plant models, so Arabidopsis and tobacco plants. And then sort of I've always had a passion for medical research. So from 2015 and onwards, I've actually looked at virus infection and viral etiology of type 1 diabetes. So that was my entry point into virus research as well as chronic diseases. And then now, since joining in 2015, I lead the virology theme within this prospective cohort study that's been ongoing for 10 years now. So we follow 1,500 mother-infant pairs across the country. They all have a high, first degree relative with type 1 diabetes. That puts them at a higher risk for type 1 diabetes than the general population. And then my role is really being to characterize the longitudinal virome dynamics during pregnancy, infancy, see if it contributes and which viruses contribute to type 1 diabetes. So the group, we're actually under the umbrella of the Virology Research Laboratory at the Prince of Wales Hospital. So I'm affiliated with New South Wales Health Pathology. And uniquely, our research lab director, Professor Bill Rollinson, he also runs and leads the virology and serology division for New South Wales Health Pathology at this major tertiary hospital. And then sort of the unique sort of setup that we have here in Randwick is that we have four major hospitals sort of joined together, listed there, and I lead two focus groups within VRL. So one's the viruses and diabetes, but also I've established sort of a human viromics group. So our group's currently, expanding. I think, this is the major key players in our group. We've got a biostatistician, Kylie Ann, who works closely with us. And then we've got a whole bunch of, you'll notice that a lot of them are bioinformaticians. So that's sort of where our efforts have gone progressively in the last 4 or 5 years. And we've got lots of PhDs and lab support as well. So I mentioned, that 1,500 mother infant pairs. This is sort of the intervals at which how we collect samples, data, and sort of do the analyses on. And that's sort of me, and it takes a whole village to run these kind of large cohort studies. And in India, we've got various themes. I mentioned virology theme is one of the key environmental factors that we think contribute to type 1 diabetes, as well as there's a big overlap between type 1 diabetes and the celiac. So we actually have a celiac theme, and among the children that we're following, there's a lot of celiac autoimmune children. So we think there's common autoimmune pathways and trigger pathways, including virus infection. So, my work really has, the first kind of work that I did was sort of virus infected human pancreatic islets. And then, if you actually infect them with an ubiquitous enterovirus called Coxsackie virus B5, that we think is diabetogenic, you lose insulin production post-infection and you get cell death. So we kind of describe that microRNA pathway, which is sort of transferring my skills from the plant microRNA work that I did. Without going into too much detail, just know that this is a thing and there's now vaccine trials and antiviral therapeutics. The first vaccine that's being developed for the prevention of type 1 diabetes will go ahead to phase 2 trial, and it's backed by Sanofi at the moment. And there's a antiviral treatment that's just finished a phase 2 randomized control trial published in Nature Medicine that shows promising results. So okay, so I mean, over the years, in the nine or so, 10 years that I've been in the sort of human virome space, we've been in the business of sort of collecting from infectious disease labs around the world, sort of best tools for the purpose of looking at the human virome. So I've listed here, you've got virus capture sequencing. So I'm calling that, so it's often also called target enrichment sequencing, and that's what you're here for, obviously, for the twist capture. And then there's obviously the metatranscriptomics, which is used a lot more by sort of friends that I have, like J.S. Eden and Eddie Holmes. This is sort of the approach that a lot of people use. And then I've put in the black box CZID. So this is actually a default workhorse pipeline for our taxonomic classification of reads that we do, and it supports both Illumina and nanopore sequencing platforms. And I know there's one codex on the line, and sort of that's also useful, and we've benchmarked in the past, but for our purposes, CZID is sort of a default pipeline. And then finally, the bottom, which I won't have time to go into, but this is an area that we've been developing in-house and kind of getting that capacity running with collaboration with Harvard, is FIPSeq. So that's actually looking at not the viral nucleic acids, but antiviral antibody repertoire, again, using NGS. So over the years I've collected, so I mentioned virome capture sequencing. We actually did we're one of the first groups in Australia. I guess we sort of adopted that back in 2016 when a publication on Roche chemistry, nimble gene capture, was published. Two papers, in fact, and one of them was from the group that Ian Lipkin was leading. So I went over and then brought that sort of virus capture, and for them it was vertebrate infectious viruses, and it was actually 2 million capture probes, and sort of brought that back into Australia. And then, as I mentioned, FIPSeq is through collaboration with Stephen Alledge and Harvard University, where they published this thing called VerScan, and it was published in Science back in 2015. And then most recently, I'm working with the TGen group in Arizona, Jason Ladner and a fellow Australian, John Elton, who's developed kind of a PipSeq like PepSeq, again, still using a PepSeq, peptide library to look at antiviral antibody presence. So these are kind of the cutting edge firing techniques that we're dealing with. And then, yeah, so the CZID came through I was an early beta tester, and then sort of I have an ongoing collaboration with Joe DeRisi, who's the director of Chan Zuckerberg Biohub in San Francisco, and he's based at UCSF. So I met him in February this year after giving an invited talk on clinical metagenomics at this hub. Um, yeah, so I won't go too much into that, but I'll just say where we have actually used the bulk and majority of twist capture and other capture tools. As I mentioned, the Roche capture, we've actually done over 1,500 stool samples from this India cohort. And then with the Twist, we actually partnered with Twist a couple of years ago to look at the respiratory samples of throat swabs in this cohort. So we've just done just under 1,500 throat swabs in that. And then with the antiviral antibodies, we've been working through the plasma. And it's been heavily funded, actually, mostly from the US and philanthropy and JDRF. Now, so I've expanding that framework where we're now moving into sort of other chronic disease areas, celiac disease, asthma, allergy, as mentioned in the introduction, and also exacerbations in CF. And this sort of idea for this ideas project actually came about from a consortium already existing and funded by the EU called the HediMed Consortium. And it's led by a close collaborator of ours, Heki Hoiti. And it's really bringing together 18 different cohort studies. So there's 22 different partners, and it's a massive, massive cohort that's also looking at environmental factors contributing to lots of different chronic diseases. So, and then sort of the international virome projects that are sort of funded now is the Horizon Europe grant, again led by Heki, and I'm leading the sort of Australian component of that from next year. And then this year, we've just participated in submission of two NIH grants, both sort of focused on the human virome. So we hope that will go ahead in the next year. Okay, so that's the research side of things. And to sort of go back to what you came here for infectious disease, well, COVID happened, as you know, and then, everyone basically working in the virology space, and particularly in the virome space, it was relevant to help with the COVID rapid response research. So it was spearheaded by Bill at UNSW, and we had this sort of and then Rowena, and you see Ira Davidson from Garvin here, and also Sebastian Van Hel from RPA. We all got together and we were trying to look at what we can do. So the key publications I've put here, one was sort of utility of nanopore for SARS-CoV-2 genome analysis that's been published, and then also what I led and looked at and was my interest was to use the twist virus. That was the respiratory virus panel, in fact, and it's the first publication from that panel use. And it was just looking at co-infections, and because there was early on worries that there would be very high percentage of co-infections, and maybe co-infections could lead to more severe outcomes, but it wasn't really the case. And fluorona kind of was a thing, but really we saw very little. So it was like 6% of co-infection rate that we found. I won't go into background of the twist because George elegantly described all that to you. But this was actually presented in another twist webinar a couple of years ago, where we actually saw that we used the original sort of Roche capture panel, which was designed, even though it had 2 million probes, it was designed prior to SARS-CoV-2, obviously. And when we compared the twist respiratory panel and looking at SARS-CoV-2 genome coverage, we could see that the twist panel obviously performed much better. And then the influenza virus, we were able to find in co-infected samples, we were able to detect full genomes across all the segments using this panel. And then again, the performance of, for SARS-CoV-2, the performance of the twist gave much better results when we're trying to correlate the CT thresholds and sort of the actual viral loads and alignments that we're getting. And then one thing I put here is that the benefits of a twist approach over an amplicon approach, this was to illustrate that. So if you actually did on the same sample, we did a nanopore amplicon sequencing as well as the twist respiratory panel, we were able to clearly, see that we're getting, even coverage all across And then there was a deletion that we found that was detected in the ORF8. But you can see that there was actually the deletion caused the primer, one of the primer A7s in the amplicon scheme, to be not there anymore. And so you had this chunk, quite a big chunk missing from the nanopore amplicon sequencing. So there's utilities when you're weighing up, amplicon versus capture. These are kind of the things to consider. If you're interested in the actual whole, pipeline, it's research use only, and it has been utilized to sort of inform diagnostics on the side. But I'm happy to share anything. So please email me or can get in touch. With the India nested case control study where we've done the respiratory virome sample, this is kind of a typical overview of what I've just selected about 100 samples that we've done on the Illumina NovaSeq X Plus. Using the TWIST comprehensive virus research panel, you see sort of a whole range of different viruses, but it's always kind of a zero sparse, it's a zero inflated sparse kind of data that you're looking at. But you would hope to see sort of usual suspects, and we do see that we see sort of rhinoviruses and a few other viruses that we hope to detect in these kind of sample types. And then just to reiterate, it's not just looking at a tiny little bit of a genome, but it's actually trying to span the entire genome of that particular virus. It is 1 times tiling for the comprehensive panel, but you can see even though there's patchiness, you get pretty good coverage across the reference genomes that we sequence. I think important factors, maybe more so if you're like me and you're trying to compare between case and control populations rather than doing, just pure detection. There's batch effects, positivity thresholds, quite arbitrary, different ways of normalizing. So if you're just doing by counts analysis in comparison, you can see that there is a particular skew and bias towards large genome size viruses. Obviously, they're going to produce a lot more viral read counts across the whole genome versus a small virus. So just kind of different ways of thinking, different ways of normalizing. All of these need to be taken into consideration. So that's actually taking us a long time. Even though we've had the data for a long time, we're currently working on these kind of things. And also the use of different bioinformatics and biostatistic tools and different assumptions, I think you need to take into consideration. And then there's also this kind of, hypothesis whether SARS-CoV-2 has been actually associated with higher incidence of type 1 diabetes or higher diagnosis rate post-pandemic. And I'm not personally a believer that SARS-CoV-2 contributes strongly to type 1 diabetes risk, but this is being looked at in India because we've been following the children throughout the pandemic. Okay, so the case studies. So one, it's very quick. It's published now in EID, which is a CDC journal. We had this unfortunate child pass away at the Children's Hospital in Randwick, and we didn't know what caused it, but we needed to know so the parents could have closure, and we also know when similar things, happen next. We all thought this was a Japanese encephalitis virus case. That's how it came to us, but it was diagnostically all negative across Westmead and Randwick. And when I used the TWIST panel to look at a brain biopsy of this child, I could detect a very unusual, pigeon paramyxovirus. So this is a virus that would otherwise be undetected. No one would think of looking at this because it usually causes mild conjunctivitis in humans, but it does wipe out whole bird populations through encephalitis. So in poultry, this is screened for routinely, but not in humans. So we described what this virus was, and since we submitted and is working to publish, there's been similar cases in China. Not fatal, but I think in this case, the child had some level of immunocompromisation. So this is work that J.S. Eden and I led. And then the second case study was an unexpected case of dengue virus encephalitis. It was a female microbiologist from Timor-Leste who came into the country and then was in and out of the hospital for various, infectious symptoms. No one knew what it was. We got a tissue, brain tissue biopsy. And then we were able to see screaming levels of dengue. So, we've done some additional validation. So, prior to this, it was hypothesized, but never really shown that you could get neurotropic dengue. But we think this is a true case, and it's currently under peer review at the moment. But yeah, screaming levels of this dengue. And then since pandemic, I've just listed how here, like George said, there's been a massive surge in clinician and public health requests, all looking at all these viruses that are not routinely screened, some of them, and sort of when you don't have a primer set established, it's something like the monkeypox, and we sequenced the second case in New South Wales through the panel. That's where I think the panel really helps, especially a comprehensive virus panel. And then there's obviously the surveillance potential. I know several groups in the world have used the, Twist or other capture platforms for wastewater surveillance. Locally, we're also looking at aircraft wastewater surveillance. And then there's norovirus outbreaks that usually are always surveilled. And then I think agriculture, animal reservoir, as you saw, if there's some kind of homology to known viruses, there is utility. But I think what I'm working more towards is actually those the antibody approach. How can we generate sort of antibody screening in these animal reservoirs? Because I think if it causes an immune response in animals, they're most likely the ones that are going to have zoonotic potential. Recognizing that I've only got 5 minutes left, I'll do very quick overview. So this is one paper that's currently accepted and impressed in pediatrics. It's been run by two sort of junior postdocs in our lab. So there's been a surge in RSV post-pandemic, as you know, and still there's RSV cases being reported, which is sort of out of season. But so one neat study that they've done is that they looked at whether this surge was, could be attributed to waning antibody mediated immunity in these cases. It doesn't seem to be the case. RSV A, B, neutralizing antibodies just stay the same across the different seasons. And then they've just described, and in the red dots is what they've been able to sequence. And this was done using the Twist respiratory virus panel. And it worked very well and got whole genome sequences quite well. And they were also able to just, characterize and see where the non-synonymous mutations were happening. And then this is the meeting that I spoke to in January. There is this major push in the US to have this common goal of FDA approval for clinical metagenomics, and Twist capture or any kind of capture approach also falls into this. And I personally think the capture approach or even syndromic panels, I think those are the ones that will get approved first and see diagnostic utility rather than a sort of pure shotgun metagenomics, because what's 10 read out of 10 million reads going to really tell you? I mean, it may be real, it may not be real, but clinicians don't want to deal with that. That's not a diagnostic. So kind of having a clear reporting dichotomous result, I think that's what really has the potential to get approved for FDA and TGA. So I'll actually skip all this. I think this is pretty generic and I think there are clinicians' attitudes and different positivity thresholds, the arbitrary nature. I think those are all kind of the barriers to actually implementing clinical metagenomics in any country and Australia. And then there's this kind of always debated approach. I still think semi-targeted or capture-based approach has the best way, but maybe updating that regularly, having a two-pronged approach where shotgun is done sort of for the surveillance routinely to update your targeted panel iteratively throughout the 6 monthly basis or something. I think that might be the best combination moving forward. So future directions, all sorts of things are planned, but I just wanted to comment very briefly that we are working to do miniaturization, automation, and we're kind of working in partnership with the Ramachadi Center for Genomics, which is an NGS provider. And really, I just want to get this, NGS accessible to the masses, and really get the cost, drive it down to a minimum. That includes capture, of course, and this is us setting it up just recently. Yeah, and I just want to really final slide is to mention that every 96 samples that we process for target enrichment, we're actually generating over 6,000 tips. So I think, one of the reasons why I invested in those robotics that I showed just before is that they're tip independent. So it actually dramatically reduces our 90% of our tip usage. But just kind of be in mind as you do any kind of NGS, let's think a bit about sustainability and how we can minimize plastic consumption. And this is, an art exhibit in EMBO in Heidelberg, and it's titled 4 Weeks of Pipetting. And this is how many pipette boxes we're putting into landfills or incinerating. So let's just think a bit more sustainable. And I think Twist does think sustainability. So I think that's a good thing. Yeah. So I'd like to acknowledge everyone and happy to take questions.